Good evening. Today I will be discussing the effect of temperature on Bacteriophage lysis against Escherichia coli. Before we get started, there are some things to know about me. My name is Tristan Hernandez, and I'm a current senior at Chatham High School. In the fall, I will attend the University of Virginia, where I plan to study mathematics. One reason why I chose this topic is because of my personal interest in microbiology. In particular, I'm interested in the study of bacteriophage, which is a class of viruses that target specific bacteria. I have long been interested in this topic after learning about it at a science program at Virginia Tech, where professors informed us of its potential use as a substitute for antibiotics. This ability may prove to be useful in the future as alternative treatment for bacterial diseases, as antibiotics are known to wane in effectiveness over time. Moreover, I'm interested in the role of bacteriophage in food safety and nutrition, as the microbe may be used to combat foodborne illnesses, specifically those originating from E. coli. In the context of microbial ecology, there has been an abundance of research done on the properties of E. coli. It is widely agreed that while stable changes in temperature over long periods of time do not significantly affect the growth of E. coli, fluctuating temperatures tend to render the bacterium more susceptible to elimination. As a result, many researchers suggest that bacteriophage can work in tandem with temperature to limit the growth of E. coli. However, there is very little consensus on the range of temperatures for which E. coli-specific bacteriophages are effective, as there has not been an adequate amount of empirical study done on thermal response. In addition, there is no clear pattern as to the effect of temperature on other bacteriophage host pairs. The conclusions can be drawn from with regards to E. coli and its bacteriophages. With regards to preventing the confounding effects of temperature on E. coli, much of the extant literature suggests that research done on bacteriophage host pairs should be conducted with the optimal bacterial growth temperature as the control. This temperature has been found by researchers to be approximately 37 degrees Celsius for the E. coli K12 strain. In addition, other studies have emphasized the importance of maintaining a steady medium that is both conducive to bacterial growth and is relatively neutral with respect to pH. In studies on bacteriophage against other strains of E. coli, many researchers have concluded that there is essentially no lysis at temperatures above 41 degrees Celsius and below 15 degrees Celsius, meaning that future research should only consider the intermediate range. Specifically, a study conducted on Listeria monocytogenes and its bacteriophages noted that using common ambient temperatures as treatment groups will maximize the applicability and interpretability of results. Additionally, many researchers have determined that the effect of temperature on a sample is mildly affected by incubation times at lower temperatures, as a precaution of future studies. Therefore, after a thorough review of the literature, it is apparent that there is a remarkable inadequacy in the coverage of different species of bacteria and bacteriophage. Furthermore, very little is known about the effect of external factors, such as temperature, on bacteriophage-host interactions. In addition, conclusions drawn from this study will prove to be a significant benefit to the field of microbiology and bacterial treatment. Considering these facts, the current study aims to address the following research question. How does temperature affect the formation of colonies by E. coli when treated with bacteriophage T4? Specifically, my study will examine temperature differences centered around the optimal bacterial growth temperature, for which my null hypothesis is if the treatment of E. coli with bacteriophage T4 is subject to a temperature lower than 37 degrees Celsius, then there will be no difference in the number of bacterial colonies formed for a sample incubated at 37 degrees Celsius. Alternatively, my research hypothesis is if the treatment of E. coli with bacteriophage T4 is subject to a temperature lower than 37 degrees Celsius, then there will be less bacterial colonies than a sample incubated at 37 degrees Celsius. I chose this hypothesis given that previous studies have shown that bacteriophage is more effective at lower temperatures. To test this hypothesis, I used incubation temperature as my independent variable. As suggested by the literature, I use common ambient temperatures of E. coli, which I chose to be 17, 25, 30, 37, and 42 degrees Celsius. In order to adequately assess the effect of these temperatures, I measured the number of bacterial colonies formed by E. coli. As a mode of control, I used the optimal temperature for E. coli of 37 degrees Celsius to compare my other treatment groups against. To provide a constant environment, each treatment group was grown in the same medium of tryptic soy agar and under the same laboratory conditions. Furthermore, every sample in the current study was handled with aseptic technique so as to not contaminate the other samples. To begin my data collection, I first gathered the necessary materials for my study. These included the following: 30 tryptic soy agar dishes, which were divided into quadrants yielding a sample size of 120, the K12 strain of E. coli, which was selected for its availability and representation of E. coli on the whole, Bacteriophage T4, a 1 to 10 microliter micropipette, and an inoculation loop and Bunsen burner. After I collected my materials, I began inoculating each petri dish quadrant with the E. coli K12 using proper aseptic technique. The top image is a picture of me inoculating the bacteria onto a dish. Then I added 5 microliters of Bacteriophage T4 to each sample using the micropipette, making sure to discard tips in between samples. The lower image pictures me using the micropipette to add this quantity of bacteriophage. At the end of inoculation and bacteriophage treatment, I taped each dish closed and incubated each treatment group at its respective temperature. It is worth noting that I originally intended to incubate each treatment group at 24 hours, which I did for the 30, 37, and 42 degrees Celsius groups. However, as cautioned by previous studies, I had to adjust the incubation times for the lower temperatures from 24 to 48 hours after no bacterial growth was initially observed at any degree. After the incubation period concluded, I began counting each bacterial growth colony for every sample. The image to the right is a picture of me collecting this data. Finally, I compiled the raw data into a spreadsheet and conducted a one-way analysis of variance on the data using the Data Classroom software. The Data Classroom software then returned this graph as a summary of my results. This graph shows the number of colonies formed for each treatment group by individual samples. Beside each data cluster, the figure graphically conveys both the mean and standard deviation of each treatment group. This graph is able to highlight many key features of the data. For instance, there are notable outliers for multiple treatment groups. In the lab, outliers designated by a colony count of 100 were those that demonstrated a complete bacterial lawn rather than isolated colonies. This condition can be exemplified by quadrant number 4 of dish number 8 as shown to the right. Although 100 may be an arbitrary value for this condition, I concluded with my advisors that this value would appropriately represent the data. Another recognizable feature is the similarity in data centers for the 30, 37, and 42 degrees Celsius groups, but a slight difference for the 17 and 25 degrees Celsius groups. This difference may be due to the change in incubation times for the lower temperatures from 24 to 48 hours. As for the differences between the 17 and 25 degrees Celsius groups, they may have been a result of the 17 degrees Celsius treatment group not being incubated for a sufficient time period, whereas the 25 degrees Celsius group may have been incubated for too long. As previously mentioned, the current study implemented the one-way analysis of variance test in order to compare the means of each treatment group. Considering the hypothesis test results, my research hypothesis that there would be an inhibitory effect of temperature on bacteriophage lysis could not be supported. All of this to say that at the 0.05 level of significance, there is not sufficient evidence to conclude that the treatment of E. coli at different temperatures yields a difference in bacterial colony formation. Although these results did not provide an affirmative outcome, this conclusion expands on the current literature for bacteriophage-host relationships. For one, these results compared to previous research on E. coli and its stressors. In that review, researchers determined that the bacterium adapts very well to adverse conditions such as temperature, so the finding of no significant difference among treatment groups may have been expected in that regard. Furthermore, the current study contrasts to the study on Listeria monocytogenes, for which there was a significant difference, by the virtue of differences in bacterial strains. This difference in classification may be reasoned to be the cause of different thermal responses. With regards to major global issues, the results of this research are evidence of bacteriophage being a viable treatment for E. coli regardless of the temperature. One glaring limitation of the current study was the practice of counting colonies as opposed to the standard practice of counting lytic plaques from bacteriophage, which are clearings in the bacterial lawn of a sample. This hindrance was largely due to the manner in which the specimen grew on the tryptic soy agar. So, given that this practice is non-standard, the results of this study may have been tainted by improper technique. And while this does limit the results of the study, it provides valuable insight into future research that this technique is not viable. In addition, the current study was limited by the continuity of the selected temperature range. Obviously, this study is unable to capture the full image of the effect of temperature on bacteriophage lysis, as the temperature values for this study, although based on reputable scientific literature, were discrete values that lack the comprehensiveness of a continuous spectrum. Future research should be able to address these limitations by making a careful effort to adhere to the standard of measuring lytic plaques in lieu of the ineffective methods of the current study. In addition, prospective research may find it beneficial to examine the behavior of bacteriophage at lower temperatures, as the current study found unique results for the 17 and 25 degrees Celsius groups. Finally, given the potential error in data collection methodology, the findings of the present study should not be overinterpreted, but future research should be conducted to expand on standard scientific practice and to aid in the development of new treatment against bacterial diseases and public health concerns. From this opportunity, I was able to not only learn of the properties of E. coli and its bacteriophages, but more broadly, I was also exposed to a field that is outside of my comfort zone. As much as I love mathematics and the physical sciences, I found it very interesting to immerse myself in the field of microbiology. Furthermore, the results of my study revealed to me that science is not always full of success and that even insignificant results are crucial to the development of new research. I would like to extend my gratitude to Ms. Long for guiding me through the research process, Mrs. Dillon for offering advice for scientific practices, and my administrators at both Piedmont Governor School and Chatham High School for accommodating data collection logistics. And with that, are there any questions? Ms. Walls?
So if I understand your process, you had the petri dish, you put the E. coli on it, and then you put the phage on it immediately after, is that correct?
Yes, that is correct.
Okay, so do you think that the experiment would have changed in any way if you had allowed the E. coli to grow during a period of time, then put the microphage on it?
Okay, so Ms. Walls is asking if the length of time between my inoculation and my bacteriophage treatment would have any effect on the outcome of this study. And based on the research that I was able to gather out of the literature that I read, most of those studies implemented the bacteriophage as soon as the E. coli was treated. So I followed suit with those studies, and I do believe that there could be an effect, but I don't think that was within the scope of this study in particular.
So at the lower temperatures, you have several outliers. Do you have any thoughts about why you had such a range there and such a difference in the number that you counted?
Okay, so Dr. Jones is asking about the prevalence of outliers in lower temperatures and how there might be a difference between the groups. To answer that, I say that the lower temperatures, I think that not having them incubated for the same period of time as the higher temperatures like the 30, 37, and 42 degrees Celsius groups, I think that that incubating them for a longer period of time adds more volatility to the sample. And so I think that might be the reason for more outliers at those lower temperatures.